Well, hello, my fellow pre-meds. Welcome back and congrats on making it to the last video of our lab techniques playlist. I know this was a short playlist, but it definitely was a dense one. And unfortunately, this video is the most dense among the 4 videos in this playlist. We will have Anki cards associated with the videos to aid with memorization, but also I'd highly encourage that you rely more on understanding the underlying mechanisms behind the concepts, because that sticks a lot longer than simply trying to memorize facts. So let's get started by discussing recombinant DNA and biotechnology. Also, as usual, there are a bunch of different lab techniques that we discuss in this video. If you want to jump to a specific one, everything is time-stamped. Okay, so just a broad overview over here. The idea behind biotechnology is that you're able to combine different DNA fragments, sequence them or read them, and then also amplify them to run different kinds of analyses on those fragments. And then the tools you have in your toolkit, first you have restriction enzymes, which we briefly introduced when we talked about blotting in the previous video. But these are essentially molecular scissors that cut DNA fragments at specific sites, as we'll see in a bit. We also use DNA ligase because when you're cutting things, you're probably eventually going to have to join things together. So we use DNA ligase as our tape over there. And remember, we saw DNA ligase in the video on DNA replication and repair in our biochemistry playlist. And finally, we have this new term, which are plasmids. So plasmids are essentially vehicles that carry foreign DNA from one cell to another to be able to clone that DNA or that specific gene. So let's get started by discussing restriction enzymes, the molecular scissors. These will cut DNA at specific sites known as restriction sites. So each restriction enzyme has a unique site, a unique nucleotide site that it's going to cut. These sites or sequences are typically called palindromes. What does it mean to be a palindrome? Well, as you can see right here, if you read the complementary strands from either side, they're essentially the same thing. So 5' GAA, and then same thing here, 5' GAA, etc. Now, once you cut the fragment, you will have 2 different kinds of ends on these fragments. What do I mean by this? You can have a sticky end and a blunt end. So you'll notice over here, if we were to cut it like that, so you go down and then to the right and then down once again, you will get these sticky ends where you have these flaps hanging out of your DNA sequence, okay? If you have the blunt end, you're essentially just slicing it in half and then getting these two fragments with blunt ends, so no flaps hanging out of them. Now, when it comes to recombinant DNA, when we're joining DNA strands together, we prefer these sticky ends. So they're the basis, really, of recombinant DNA. And we're going to take a look at how recombinant DNA works. It's a very simple concept. You can take a look at it over here. So you want to cut out this fragment and add it over here. You get your restriction enzymes. You cut out this part and you cut out this part. So now you have your fragment that you want to add to the new DNA strand, and then you just transfer it over. So it's that simple. Now this is recombinant DNA. So now we're going to move on and apply it to plasmids, where not only can you add it to a new DNA strand or new DNA circular DNA strand over here, but then you can move it somewhere to a different cell with plasmids. So the idea here is that plasmids are circular double-stranded DNA that exist in bacteria. Importantly, they're independent of the main chromosomal DNA. So recall, bacteria contain this circular DNA as their genome. This is like a small DNA sample, a small circular DNA that is separate from the main genome. Now, key features when it comes to plasmids is that they contain restriction sites. So it's important that they contain these restriction sites so that you're able to cut the restriction sites and incorporate new DNA so that you can form recombinant DNA and clone your genes, okay? And they also contain this ORI site, is short for origin. Remember, the origin of replication is what allows the DNA double helix to replicate, or in this case, the circular DNA. Of course, this is also double helix, but it's just circular. Allows it to replicate, so it can replicate independently because it has that origin of replication site. Finally, it'll have this antibiotics resistance gene. So this is a selectable marker, and we'll see what the purpose of this is in just a bit. But just remember that you need that antibiotic resistance gene to be there. So let's talk about how we recombine these plasmids. Essentially, it's the same thing as we just saw. Let's say we have this plasmid over here, and then we have this DNA from the human genome or from like an animal cell that contains the insulin gene, the gene that's eventually going to express insulin and eventually be translated into insulin. So what you do first is you get your molecular scissors, your restriction enzymes, and you've designed these previously, or you bought them from a lab that designed them specifically for this gene. So they recognize the specific sites for this gene. And you cut that part out right there. And you also allow your restriction enzymes to cut the plasmid over here. So then you can kind of join the gene that encodes for insulin onto the plasmid. You're going to anneal them so they get close to one another. But then you need ligase to come and ligate them and basically like seal the gaps over there. So now you have your plasmid that contains the gene that transcribes for insulin that will eventually be translated into insulin. But you do technically run into an issue over here, and we're going to see how we're going to solve it in just a bit. But let's just talk about the issue that arises here. Recall that eukaryotic DNA, so DNA that you got from an animal cell or from the human genome, in this case the insulin gene, is not only going to contain the region that's eventually going to transcribe into mRNA that can be translated into protein, but it's also going to contain introns. The introns should not be in the mature mRNA that is translated into proteins. Now, luckily for us in eukaryotic cells, we have the machinery to be able to cut out the introns and leave the exons, which will be translated into protein. But of course, this process in eukaryotic cells occurs after you transcribe the gene. So you transcribe the gene, contains both introns and exons, then you get your spliceosomes, the machinery that can cut out the introns and leave only the exons. But if we just got the insulin gene from, say, a human cell and transferred it to a plasmid, which we're going to put in a bacterial cell, well, that gene contains both introns and exons. So when the bacteria transcribes the gene, it's going to get mRNA that contains both the introns and the exons, and therefore will not be able to translate it into the insulin protein, the protein of interest. Keep that issue in the back of your head, and we're going to see how we're going to solve it in a bit. But let's get back to the plasmid right here. So we've got our fully functional plasmid, we got our fragment of interest, we got our antibiotics resistance gene, and the origin site. So now we apply the plasmid onto a bacterial colony, and of course, some bacteria are going to uptake the plasmid while others won't. So here we can see this one took the plasmid in, whereas these didn't. But you can't just look at a microscope, at the bacteria under a microscope, and determine, okay, this one got the plasmid, this one didn't. So what's a better way? Well, since we have an antibiotic resistance gene over here, this one will be able to survive in an environment that contains an antibiotic, say for example ampicillin. So what you do is you get a petri dish and you cover it with ampicillin, and then you allow your colony to grow over there. And you're essentially creating a survival of the fittest, evolutionary, like evolution in real time type of situation, where only the bacteria that contain your plasmid of interest will survive and thrive in this petri dish. The rest will die out. So now you're guaranteed to only have the bacteria that contain the plasmid. Okay, now let's talk about how we solve the problem of our gene fragment containing both introns and exons. Well, we solve it with what we call complementary DNA. Complementary DNA is a single-stranded DNA strand that's been synthesized from an mRNA strand. So we're reversing the central dogma using this enzyme called reverse transcriptase. So recall, the central dogma goes from DNA to RNA to protein. Here we're going from RNA to DNA using an enzyme which we'll see again and again in the biology playlist when we talk about viruses, using this enzyme called reverse transcriptase, which uses an mRNA strand to synthesize a single-stranded DNA strand. Now, what's the point of making this cDNA, or what we call complementary DNA? Well, the purpose is that now this DNA segment, this DNA sequence, only contains the coding regions, the exons of your gene, right? Because it's based on the mRNA sequence. So let's think about how we got this mRNA sequence. We started off with the insulin gene, we transcribed it in a eukaryotic cell. So you got your mRNA that contains both introns and exons, and then the spliceosome cut out the introns and you're left with the exons. So this mRNA strand only contains the exons that can be translated into protein, okay? So when you reverse transcript it, so now you get your ssDNA, this only contains the coding regions, the exons of your gene. So now you've solved the problem. Of course, you allow it to form double-stranded DNA with DNA polymerase, but you've solved the problem. Now, if I get this double-stranded DNA and incorporate it back into my plasmid over here, the bacteria does not have to worry about the introns and exons, because the gene only contains the exons in this case, so it'll transcribe the gene and then be able to translate it into the protein of interest. And just as a side point, this was indeed used to synthesize a bunch of insulin. So you inject these plasmids into bacteria and you allow them to do your work, you make insulin, and then you can use that insulin for medical purposes. Also, one last thing when it comes to this genomic versus cDNA library, not entirely high yield on the MCAT, but the idea is that if I wanted to get an idea of what genes exist within a cell, I can sort of cut up the genome and then analyze the different genes, and that would tell me exactly what genes exist in a cell. So like, let's say there are 1,000 genes in that cell, you'll be able to identify those 1,000 genes. But what if I'm only interested in the genes that are actively being expressed? Like, I don't care about the silenced genes. I only want to know what's currently being expressed, transcribed into mRNA, and then translated into protein. Well, if I use a cDNA library where I use reverse transcriptase to transcribe cDNA from the mRNA transcripts, then I know that any cDNAs that I get over there represent the genes that are actively being expressed in the cell. Okay, now let's move on to polymerase chain reaction. Very high yield on the MCAT and very revolutionizing technology, not only in the field of medicine, but also in the field of forensics. Like, you had people who were previously convicted of crimes for years, if not decades, that were actually innocent, but obviously with our flawed system, they were put in jail for decades in some cases. Once we were able to develop this sort of technology and other means of biotechnology, they were exonerated and found innocent after decades. So truly revolutionizing technology. So the idea behind PCR is that you have a fragment of DNA and you want to amplify it. So if you have like one piece of that DNA or 20 copies or 30 copies, you're not really going to be able to do much with those. You can't really analyze them. But if you can amplify the fragments to, say, 1 billion copies, then you can really analyze them in a better manner. So let's see how it works. The raw material that you're going to need, first of all, you need your DNA sample, of course. Then you need DNA polymerase, because you're going to be replicating DNA. And then you need deoxynucleotide triphosphate. That's just the nucleotides that are going to be incorporated into DNA. Since you're replicating DNA, you need the building blocks. And finally, you need primers. So why do we need primers? Well, recall that DNA polymerase cannot just start synthesizing DNA from scratch. It needs someone to come and lay down those first building blocks for it so that it can continue the process of replication. And then in the DNA repair and replication video in our biochemistry slide deck, we talked about primase laying down those first building blocks. Well, here we sort of cut the middleman and just design the primers ourselves and then allow DNA polymerase to do its job. So there are 3 steps when it comes to the polymerase chain reaction, or PCR. The first step is denaturation. So you raise the temperature, and the purpose of this is not so that you break the DNA, you just separate the 2 strands. So when you raise the temperature, you separate the 2 strands like such. The next step is you're going to cool down the solution a bit so that you have the annealing phase. The annealing phase allows for the primers to stick onto your DNA strands. You're now single-stranded DNA. Okay, and importantly, one thing about primer design, because sometimes they'll ask you how to design primers, the idea is that you want to make sure that your primers run from 5' to 3'. So this end where you're going to be adding your new strand should be the 3' end, because DNA polymerase can indeed only add new nucleotides to the 3' end. So for example, they'll give you this sequence, let's say it's 3' AAT, then your primer should be 5' TT A3', and then DNA polymerase can do its job. So the next step, as you may have predicted, you warm up your sample a bit so that it's optimal for DNA polymerase to do its job, and it'll start the extension process where it synthesizes the rest of the DNA strand. Okay, and just as an aside, like if you actually did this in the lab, you're not the one that's heating, cooling, and heating. That was in the past. It was like that. You had to basically get 3 water baths and put your solution in the hot one, then the cool one, and then the warmer one, and so on. Now it all happens in just one machine. The machine will raise the temperature, lower the temperature for you. So a lot easier to do nowadays. All right, one final point over here. With each cycle, you are doubling the amount of DNA that you have. So like, let's say the first one you had 1 sample, next you'll have 2, then 4, then 8, and so on. And just after 30 cycles, because you're going 2 to the nth power, where n represents the number of cycles, so 2 to the 30th power, you will be at 1 billion copies of DNA. I know this is a bit counterintuitive, but because of the exponential growth, you will indeed reach 1 billion, and you can try to do this on your calculator. But you can trust me, it's going to reach 1 billion copies just by 30 cycles. Okay, now let's talk about some offshoots of DNA, of polymerase chain reaction that show up on the MCAT. So you have what's called reverse transcriptase PCR. So again, we're seeing this reverse transcriptase. So now we're probably dealing with RNA, and that is indeed the case. So here you can measure, detect, and identify RNA sequences within your sample by using reverse transcriptase PCR. So it's going to amplify, so it's going to synthesize cDNA and then amplify your sample, right? And the idea here is that you can study RNA viruses. For example, in the COVID-19 pandemic, we used PCR to detect RNA within patient samples because the COVID-19 virus is indeed an RNA virus. You can also use this technique to check whether a gene is being transcribed or expressed. Recall from our previous video, we talked about how if you want to study gene expression, you can use Western blotting or Northern blotting. You can't use Southern blotting because Western studies proteins. So if the protein is there, then the gene is being expressed. And then Northern blotting studies RNA. So if the RNA transcript is there, then the gene is being expressed. But if you just look at Southern blotting, then you're looking at DNA. So it's not necessarily being expressed, right? Well, this is another technique you can use to study the expression of genes. Reverse transcriptase PCR, shorthand RT-PCR. The other offshoot is quantitative PCR or real-time PCR. Don't get these mixed. So real-time PCR is the same thing as quantitative PCR, abbreviated qPCR. So you don't abbreviate real-time PCR, RT-PCR. That's reverse transcriptase PCR. And now the difference between reverse transcriptase PCR and quantitative PCR is that RT-PCR will identify whether the RNA sequence is present and what RNA sequence is present, but it won't tell you the concentration in real time. You need quantitative PCR to be able to identify the exact concentration of RNA transcripts within your solution. Okay, so now let's move on to reading DNA sequences. So now you have a sequence and you wish to identify the exact order of nucleotides within that sequence. So like 5' A, T, C, G, G, etc. Okay, it starts off kind of similar to PCR. So let's look at the raw material over here. You have your DNA sample. So this is an unknown sample. You don't know the sequence and you want to know what the sequence is. You also need DNA polymerase because we're going to be replicating the DNA. You need primers because, again, DNA polymerase will not start replicating on its own. And then you need deoxynucleotide triphosphate, just your regular nucleotides that are incorporated into DNA. But then you need these special type of nucleotides called labeled DDNTPs. What does it mean to be a DDNTP? Dideoxynucleotide triphosphate. Let's take a look at what this means. So here we have a nucleotide triphosphate. Notice we have an OH at the 2' and the 3' sugar right here and right here. So this one is what's incorporated into RNA. This specifically is adenine triphosphate or adenosine triphosphate. Next we have DNTP or deoxynucleotide triphosphate. Notice we are missing the OH on the 2' carbon, and that's why we call it deoxynucleotide triphosphate. Well, when you're missing the OH on both the 2' and the 3' carbons, now we call it dideoxynucleotide triphosphate. Why is this significant? Well, because if we look at the backbone of DNA, the way we add new nucleotides is by adding them to the 3' end right here. And if you lack the OH at the 3' end, you are unable to continue the synthesis of DNA. So notice both the NTP and the DNTP still contain that 3' OH, and that's why you're able to synthesize RNA and DNA. But if you lose that OH on the 3' end, you are not able to add any more nucleotides. So the moment you incorporate this type of nucleotide, the DDNTP, you terminate replication. And this is the exact underlying mechanism behind Sanger sequencing. So let's go back to the procedure over here. So The difference starts right here, where instead of having one tube in PCR where we mix everything in one tube, here we have 4 separate tubes. Each of the tubes is going to contain all of these, okay? So DNA sample, DNA polymerase, primers, DNTPs, and importantly, each of these tubes, tube A, C, T, and G, are going to contain all the regular nucleotides, so all the DNTPs, so DATP, DTTP, DCTP, and DGTP, all of those are going to be present over here. And moving forward, just so I don't trip up over my words, I'm going to say regular nucleotides for DNTPs, and then the irregular nucleotides for DDNTPs. So for the regular nucleotides, this tube contains all of the regular nucleotides, including adenine, cytosine, thymine, and guanine. Whereas for the irregular nucleotides, it only contains the DDATP. Likewise, this will contain all the regular nucleotides, but only the irregular cytosine-based nucleotide, and so on, okay? So now let's see how this actually works. I know this is a lot. I'm gonna walk you through it. Essentially, with Sanger sequence, as I mentioned earlier, we're gonna start off by separating. We're going to start off very similarly to PCR. We're going to separate our 2 strands. So now you have 2 single strands, and you allow your primers to anneal onto your single strands, and then you allow DNA polymerase to start synthesizing new DNA. Now, again, we have 4 separate tubes. Here I only represented 3 of them. So we have the T tube, the C tube, the G tube, and of course there will be an A tube. Let's take a look at what happens in the T tube. As soon as you encounter an adenine, of course, what are you going to add? You're going to add a thymine-based nucleotide, right? So we have 2 different thymine-based nucleotides in the T tube. We have the regular thymine nucleotide and the irregular, the DDNTP nucleotide. And what's going to happen is that your DNA polymerase will sort of randomly incorporate either one. And indeed, you're going to have both of them being incorporated. If the irregular one is incorporated, then you terminate replication, and all you have is one nucleotide, this first thymine. Don't worry about the primer. The primer isn't part of the sequence of interest, so this is just the very first nucleotide. Okay, but if you get the regular nucleotide, the regular thymine nucleotide, then it allows for the continuation of replication. And then indeed, once again, you will encounter an adenine. And over here, you have the same dilemma. You're either going to get the irregular or the regular nucleotide. If you get the irregular one, you terminate replication, and you get a fragment that is now made up of 1, 2, 3, 4, 5 nucleotides, right? So what's going to happen is, by the end of this, you will have every possible length of fragments that terminate with a thymine. Of course, here, when we incorporated the regular thymine, why were you able to continue synthesis? Because we mentioned that every single tube has all of the regular nucleotides. So that's why you're able to continue replication. But importantly, as I mentioned, by the end of this process, in the T tube, you'll have every single fragment that terminates with T, and that fragment that terminates with T, each one will be labeled, so you'll be able to detect it. And then the same thing will happen in the C tube, in the G tube, and so on in the A tube. So even if you didn't understand any of this, because I know it is a bit tricky, even if you didn't understand it, you can still answer questions correctly. But having a solid understanding will really allow you to answer the questions more efficiently. But again, we'll see how even if you don't have a solid understanding, you can still answer questions. So now what we're going to do is we're going to transfer our samples via gel electrophoresis, and we're going to separate them. So remember, gel electrophoresis, this is the negative side, and this is the positive side. So this is a positively charged anode, so the fragments are going to be attracted, because DNA fragments contain negative charges, are going to be attracted towards that positive electrode, and the bigger they are, the less they're going to travel, right? The smaller they are, the more they're going to travel. So you look at this very last fragment over here, you can deduce that this one must be my very first nucleotide, because it's the lightest fragment. So my very first nucleotide in my sequence is going to be thymine. Well, the next one, which one's the next lightest, is going to be this one. This one contains AT, so I know that the next one is going to be A. This one is going to contain CAT. That's why it's a bit heavier than those ones, and so on and so forth. So from the bottom to the top, you can read 5' to 3'. So it'll be 5' T, A, C, G, etc. And that's what we have here, 5' T, A, C, G, etc. So even if you don't understand the mechanism, as you can see, if you just know this, you'll be able to answer most questions when it comes to Sanger sequence. But there's one twist over here, that this is actually not your sequence of interest. This is what's complementary to your sequence of interest. Because notice what happened over here. When you, let's say this is our strand of interest. This is the strand that encodes for insulin, for example, and you wanted to see what the exact sequence of nucleotides are within it. So you heated the sample up and you denatured it, and then you allowed DNA polymerase to carry out its function. Well, the new strand that it's making, and then you're going to read that strand, is going to be complementary to your gene of interest. So if they're asking you about the gene of interest and not the gene that's complementary to it, then you need to first sequence your sample and then take what is complementary to it. Hope this makes sense. I know there's a bit of a tricky concept, and that's why I purposefully included an example right here. I usually don't like to include questions, as I mentioned earlier in the playlist. I don't like to include questions within this playlist because I want to try to keep it as concise as possible. I'll have separate videos where we go over exercises and sample questions, but let's go over this one. A researcher isolates part of the insulin gene from the human genome, then sequences it using the Sanger sequence technique, so the technique we're talking about. The gel is shown below. Which of the following represents the sample that the researcher isolated? Okay, so let's sequence this real quick. And this would be C, G, so of course 5' C, G, A, C, G, etc., etc. Okay, so that is our sequence right there. Now, importantly, this is not the sample of interest. This is not the insulin gene. This is what's complementary to it. So I'll have to take what is complementary to this, which is 5' T, G, C. So if you immediately looked at the answer choices, and just based on this, you would have chosen answer choice A, but you want to make sure that you take what's complementary to it and choose answer choice B. And that is indeed the correct answer. Next, from here on out, everything that we're going to discuss is not as high yield of what we discussed thus far. So this is a lot less high yield, but I think it's worth understanding for the MCAT. So let's talk about DNA fingerprinting, RFLP, and STR analysis. So essentially, RFLP and STR analysis are both kind of fingerprints, like unique markers that we have in our genome that we can identify, for example, for forensic purposes or paternity tests. And RFLP, known as restriction fragment length polymorphisms, are sort of the more traditional way. Now forensics are really moving towards STR, so that's probably more relevant. But let's start with restriction fragment length polymorphism. The idea is, in the non-coding regions of your genome, you have these nucleotides that'll essentially just repeat a bunch of times. So there'll be like 8 to 10 to like 12 nucleotides, and they'll just continuously repeat. So in this case, I just have an example right here. You have A, C, G, T, A, C, C, T. So these are 8 nucleotides, and they appear to repeat 20 times in sequence, okay? So this total fragment would be 160 base pairs versus this other one, which is made up of 10 nucleotides, repeats 5 times, so the total fragment is made up of 50 base pairs. And you can see this represented over here. So this one's a larger fragment, for example. It's probably the one that's 160 base pairs. This one's a shorter one, so it's probably the one that is 50 base pairs. And what you're going to do is, why we call it restriction fragments, is because you can cut them out with restriction enzymes and then use them to determine, for example, a paternity test. We'll see this right now. So the idea is that children will inherit one of these from mom and one from dad. So take a look at this gel electrophoresis over here. So parent number 1 has this very heavy fragment, and that's probably this really long one. And then they have this kind of intermediate one right here. And then parent number 2 has this very short one. That's why it migrated the farthest. And then they have this kind of intermediate one right here. And you can see that child number 1 and child number 2 both inherited this really long one from parent number 1. But when it comes to parent number 2 gave different fragments to different children. So the very light one ended up with child number 2, and the kind of intermediate one ended up with child number 1. Now, importantly, if these were identical twins, what you'll see is that they will have both fragments will be identical. So this one would probably be up here, or this one would be down here if they were identical twins, because they'll have the same genome. Okay, not very high yield on the MCAT. I think STR, although even STR is not very high yield, I think it's a bit more high yield. So STRs are also in non-coding DNA sequences. Instead of being like 8 to 10 base pairs long, these are more like 2 to 6 base pairs long, and they repeat a bunch of times. So, for example, we can use this in paternity tests, and we'll see, we have an example right here. We're going to determine if this person is the father. So let's see how this works. So essentially you get these repeating sequences, and you're going to use PCR to amplify the sequences, and then you can run a gel to identify for example, a paternity test or forensics analysis. So let's take a look at a sequence right here. You have 5' AGT 3', and this appears to repeat 100 times. So it's going to be 300 base pairs long. And again, this is like unique to this person. Like no one else will have the exact number and the exact sequence of nucleotides, right? Because you can have like 300, 303, 306, or maybe 200 if you have just 2 base pairs repeating. Or 4 base pairs repeating, you can have 200 or 400. So there's so many different combinations to the point where it truly is unique to every individual, right? So this is going to be 300 base pairs long. This other one for the father, the potential father, is going to be 200 base pairs long. So we see that represented here. The mother has 150 base pairs long and 450 base pairs long. So again, the 450 is probably this one because it's the heaviest fragment, and this is the 150 because it's the lightest fragment, so it migrated the farthest. And because the child is going to get one from mom and one from dad, we can determine whether this person is the father or not. So of course, they got one from their mom over here. Let's see if they got one from their dad. Moment of truth. Drum roll, please. This is the father, okay? Because these are at the same exact level, okay? So that person is the father. Okay, let's quickly apply this to forensics. So we have DNA found at a crime scene, and we want to see suspect 1 and suspect 2 if they match that DNA. So suspect 1 is innocent. They don't match the DNA. And suspect 2 is also innocent. I know these appear to be close, but they're actually far apart. And plus, they'll have to have both fragments, right? Because if it's their DNA, they should have both fragments at the same level, okay? And so both of these people are innocent, as are many people who were found innocent after the advancement of biotechnology, PCR, and yeah, just biotechnology in general. Okay, now let's talk about microarray. This one shows up on the MCAT, DNA hybridization. So what you have here is essentially you're comparing between a normal cell and a cancerous cell. And you want to see, of course, it doesn't have to be a normal versus cancerous cell. It can be just any 2 cells. But what you're trying to see is which genes are being expressed more in cell A versus cell B. Here, we're specifically looking at a normal versus cancerous cell. So in a cancerous cell, some genes may be overexpressed, others might be underexpressed. Still, some will be unchanged. They'll be expressed at the same level. So let's take a look at our genes right here. We have gene 1, 2, 3, and 4, and 5. You can see that gene 1 is being overexpressed in the cancerous cell. Gene 2 is being underexpressed. Gene 3 is normal. Genes 4 and 5 are overexpressed. So these little fragments here you see within the cells are RNA fragments, RNA strands that indicate that genes are being transcribed and therefore expressed. So what you're going to do is first you're going to label all your mRNA samples in both cells with different colors. So the normal cell, we have it in red over here, and the cancerous cell, we have it in blue. You don't have to do red and blue, and you won't only see red and blue on test day, but that's just the colors that I chose. Okay, so what you're going to do is you're going to get your microchip over here. So this is like very, very tiny. That's why we call it microarray. And what you're going to do is you're going to fill these wells. Of course, they're very tiny, but let's just call them wells. You're going to coat them with cDNA that's going to be complementary to the mRNA from gene number 1. Okay, and that's because it's in this first well over here. Okay, so this is cDNA that's complementary to whatever mRNA strand gene 1 expressed. So you're going to add your 2 samples from the normal cell and the cancer cell at the same time over here, and whichever one is present in greater concentration, well, that's the color you're going to be able to detect. So in this case, because we have more gene 1 being expressed to a greater extent in the cancerous cell, you're going to see in the first well, you're going to see a blue color, okay? And that's what you see right there. Next, we get in the second well, same thing. We code it with complementary DNA to whatever mRNA sequence that gene 2 is expressing. And because the cancerous cell underexpresses gene number 2, you're probably going to see more of the red color, and therefore well number 2 or dot number 2 will be red. All right, moving on to gene number 3, where you have no change in the expression of the gene between the cancerous and the normal cell. So you're probably going to have a mixture, right? And that's exactly what's going to happen. You're going to have both red and blue dots. So the combination of that is going to be a purple dot. You're going to see a purple dot. So in summary, blue, in this case, indicates increased expression. Red indicates decreased expression in the cancer cell relative to the normal cell. And then purple indicates no significant change in expression. Of course, you didn't have to use these colors. You could have, for example, used yellow here and blue here, and then the mixture of those would be green. So if there's no change, you would see a green color, okay? Now, let us move on to some other techniques, and these are really low yield on the MCAT, but I thought it's worth briefly mentioning them. So you have in situ hybridization, called ISH, or sometimes FISH, because we have fluorescence in situ hybridization. This detects DNA or RNA sequences within intact cells and tissues. So recall from the blotting techniques, you had to kind of lyse your cells and extract your DNA in order to run them on the gel. Here, if you want to identify a DNA or RNA sequence within an intact cell or intact tissue, you can use this technique and light it up while the cell is still intact. Like, you don't have to break it down. Okay, and then immunohistochemistry is the same thing essentially, just the counterpart of Western blotting. So Western blotting, we were looking for proteins, and we had to lyse the cell, break the cell down to extract those proteins. Well, here you can detect using antibodies while the cell is still alive and not broken down. Okay, and then flow cytometry is used to identify markers on cells, identify the size of cells, and different characteristics of cells. So the idea is, for example, it shows up frequently, not on the MCAT, but like if it will show up, it will show up like you'll have like, for example, T cells or B cells. They have specific markers, and you can use those markers to identify what your cell population are. And of course, T cells and B cells from our immunology video in the biology playlist. We'll talk more about those over there. Okay, again, this is also not very high yield, but it can show up, and it's pretty straightforward, so it's worth learning. So if you want to determine the function of a gene, you can use one of several different methods. So the very first thing, the most straightforward thing, is knock out the gene. So if I have a gene, let's say this gene with a question mark over here, and I wanted to see, okay, what is its job? Well, I can get two mice, and let's say these mice are siblings. I'll knock out the gene in one, and I won't in the other. And I'll see what happens to the mouse that I knocked that gene out in. I noticed that the mouse kind of changed color over there, so I can deduce from that that this gene was probably involved in determining the color of the mouse, okay? Next, you can use a technique known as evolutionary comparison or homology between the sequences of DNA. So the idea here is you get your gene of interest that you're trying to determine what its function is. You use Sanger sequencing and then identify its sequence. And then you see other genes which you already know what their function is, and you see if there are similarities in the sequence between the two genes. And the more the homology, the more that DNA sample is conserved between the two, the more similar they are, then you can deduce that they probably share a similar function, right? And now let's move on to the next technique you can use to determine a gene's function. You can light up its protein, so the product of it, what it's going to eventually translate into. You add this marker called GFP, which will light up. It's fluorescent, and you'll be able to see where that protein will end up. So does it end up in the nucleus, in the membrane, in the cytoplasm? And from there, you can sort of deduce what the function of it is. Finally, you can see binding partners of that specific protein. So the outcome, the protein that was resulted from that gene, you can see, is it going to interact with other proteins? Is it going to interact with lipids? And based on that, you can also uncover a bit of what the role of that gene is. Okay, finally, this last topic over here, while you don't have to necessarily memorize anything over here, just keep these in the back of your mind because they will sometimes show up in passages. The safety and ethics of DNA and biotechnology. Of course, some of the concerns, number one is privacy. You know, employers, insurers, and organizations and governments can misuse the information, the genetic information from individuals. So we want to make sure that that is private information and really restricted confidential information. Gene editing, so think about what happens, like the implications if I were to edit a somatic cell in my body versus a germline cell in my body. If I were to edit a somatic cell in my body, it's only going to affect me, right? Because I'm not going to pass it on to any offspring. But if you were to edit a germline cell, that's going to be passed on to your offspring. And well, that kind of raises an ethical dilemma. Should you be able to do that? When it comes to GMOs, of course, there are upsides and downsides to every technology. I know GMOs get a really bad rep, but you want to weigh the risks and benefits of everything. So that's a consideration. And then another thing is genetic enhancement, what are sometimes called designer babies. So we might be able to agree that using gene editing techniques such as CRISPR, which you don't have to know the details of for the MCAT, but for diseases, for certain genetic diseases, probably ethical to use. But then what if it's just for the sake of changing your child's hair color or changing your offspring's eye color or things like that? Should you be able to do that? Or, for example, if we were able to identify a specific gene that's associated with intelligence or plays a role in intelligence, should we be able to edit that? And what kind of disparities does that raise between people who are able to access that technology and others who aren't? Okay. Finally, we have biosafety. So we really need to regulate. So for example, some labs may be creating certain organisms to test different things. So editing the gene pool within certain organisms, certain colonies of bacteria, for example. So we just really need to regulate those type of labs and activities. All right, well, congrats to you all on concluding and finishing the last video of the Lab Techniques playlist. I really hope you found it beneficial. If you did, I'd appreciate it if you share it with your friends, subscribe, and check out my other playlists. I'll see you in the next video. These videos, at least as of now, are sponsored by you. If you wanted to support me via Buy Me a Coffee or Patreon, you can find the links in the description box, as well as the links for everything else. Thanks for watching, and I'll see you in the next video.